next and the last speak before lunch is by Professor Susannah Porter from the University of California at Santa Barbara. And her research specialty is on the evolution of eukaryotes and its relationship to environmental changes. Yeah, there's the title. The audience is yours.
All right, thank you. All right, great, thank you. Well, what a pleasure it is to be here today. Thank you all for coming, and what a pleasure it is in particular to be here to honor Andy Knoll. It was quite a privilege for me to be a student in his lab. As I was saying earlier to Andy, I feel like it was sort of the golden age of the Knoll lab with all of my academic siblings and the postdocs in there as well. So today, I'm going to talk about the rise of eukaryotes. And this is focused on basically the early fossil record of eukaryotes. And I'm going to talk a little bit about what I think maybe led to their rise to prominence starting around 1 billion years ago. And this talk is going to form a sort of a prelude to Shuhai's talk later about early animals and Nick Butterfield's talk about the appearance of eukaryotes in animals and how that affected the environment. Now this slide shows just a sampling of the stunning array of eukaryotic organisms that are around today. Eukaryotes share in common the possession of relatively complex cells with DNA packaged inside the nucleus, with a relatively complex cytoskeleton, which allows the cell to change shape and move in some cases, and various organelles, such as the mitochondria, where most of the cell's energy is generated. Eukaryotes include the animals and the land plants and fungi and a variety of seaweeds, but they also include a great diversity of single-celled forms known as the protists. And though protists have traditionally been understood or described as merely being types of tiny animals or tiny plants, work over the last few decades has shown that actually eukaryotic diversity primarily lies within this protist group. As you can see in this unrooted tree here, most of those lineages are protistan. And the animals and the plants and the fungi, which I've highlighted here in the red circles, are sort of latecomers. They are highly derived just in the crown of the eukaryotic tree. And so it's actually more accurate to think of protists not as tiny animals and plants, but rather as animals, plants, and fungi, as just highly unusual, highly successful protists. Protists are much more interesting than we usually give them credit for. They're often surprisingly complex morphologically. For example, this dinoflagellate, which has a camera-type eye composed of a variety of subcellular structures that are analogous to the cornea and a lens, a retina, and an iris. Their behavior can be fairly complex as well. For example, these choanoflagellates here, you can see have these finger-like extensions of the cell, which it uses to weave baskets of glass strips. Or this testate amoeba here, which builds its shell out of mineralized scales that it has stolen from the test of its prey. Protists also, like humans, can adapt their local environment to live in places that otherwise wouldn't be hospitable. So, for example, this colonial ciliate, which lives in sulfide-rich habitats, which normally would be toxic, but it does so by hosting bacterial symbionts that detoxify the local environment. And there's an added bonus in that it uses these ectosymbionts as food as well. So you can kind of think of them as little farmers that are taking care of their livestock. So it's easy to fall in love with this group, the protists, as I did when I was a grad student working with Andy Knoll. And of course, since I'm going to talk about the early fossil record of eukaryotes, it really is a fossil record of early protists. Before I get into that, I just wanted to briefly acknowledge how much we have learned about the early fossil record of life and eukaryotes in particular during the course of Andy's career, and much of it from Andy's work and those of his students. And I'm going to start with Darwin, not because Andy's career started in the mid-19th century, but because it's a useful starting point for you to see the extent of acceleration that has happened in the last few decades. So you're all familiar with the struggle that Darwin had with the fossil record, right? He recognized that, he has a variety of species. He says species belonging to several of the main divisions of the animal kingdom, they suddenly appear in the lowest known fossiliferous rocks. And Darwin recognized that if his theory were true, then there had to have been a long period of time when the ancestors of these lineages must have evolved, and yet they're not showing up at all in the fossil record as far as he knew. So it was a real conundrum. If we fast forward almost 100 years, it's pretty much the same story. So at that time, the oldest widely accepted evidence of life is basically latest Precambrian in age. And then fast forward to today, so the last 70 years or so, we have a record going all the way back to basically as far as the oldest rocks that are capable of preserving life go. So here's sort of in broad strokes, we now know that life is extremely old, that the prokaryotes, bacteria and archaea, diversify metabolically and ecologically and spread across the globe through the Archean and the Paleoproterozoic. That eukaryotes had appeared at least by, say, 1700, 1600 million years ago, and they diversify. So that's the record of the protists. And then the animals, which Shuhai will touch on, are still, appearing just before the Cambrian. And it's amazing how much of this knowledge came out of the work of Andy Knoll and his students and postdocs. And just to give you a brief example, this is a compilation from the work of Phoebe Cohen and Francis MacDonald, looking at the diversity of eukaryotes in a variety of assemblages, 59 assemblages, sort of laid out here from the beginning of the Mesoproterozoic, 1600 million years ago, to the end of the Ediacaran period, 539 million years ago. And I was curious to see what would happen if I removed every study that either Andy was an author on or a study that was done by one of his students or postdocs, and this is what it looks like. So in my lab, we joke about the Andy Knoll effect, the extent that one person's contribution to this record maybe could be skewing our sense of diversity through time. Andy worked a lot on the Tonian, and maybe if we take Andy's work away, there's a very different picture of diversity. Let's see. So I am going to go, I should mention, as I go through this talk, I'm going to highlight those studies where an Andy Knoll offspring was involved, and as you'll see, it's most of the slides. So let's go back to the story of this talk, which is the early evolution of eukaryotes and their diversification. The oldest widely accepted evidence for early eukaryotes is a variety of fossils that appear in rocks starting about 1.8, 1.7, 0.6 billion years ago. These are all organic in composition, and they're microscopic. They're typically about 1/10 of a millimeter or less in size, so the size of a grain of dust or even smaller. And they're part of this larger group, collectively known as the organic walled microfossils. Now, the organic walled microfossils actually go back much earlier, to 3.2 billion years or more, but those earlier ones are all simple, they're smooth-walled, and we don't really know where they could very well be accommodated within the prokaryotes. So these fossils here are the first time that we see organic walled microfossils that show structures that are reasonably interpreted as indicating the presence of eukaryotes. And in particular, Fossils like Topania plana have these spines, in some case branching. You can see Scheuosporidium as well, hollow spines that are sort of most reasonably explained as being the result of these finger-like extensions of the cell, the ones that you saw with the choanoflagellate weaving its baskets, those involved in forming these structures. So that suggests the presence of a complex cytoskeleton. There are also fossils, like you can see here in Scheuosporidium and Satka favosa, where there appear to be plates forming the wall. And this has been interpreted to, these have been thought to be formed intracellularly and then transported to the periphery of the cell and put in place. And if that's true, that is also consistent with a complex cytoskeleton and things like an endoplasmic reticulum, characteristics of eukaryotes. So these forms that I just showed you, they persist through the next several hundreds of millions of years, some cases even later. But what tends to be characteristic of the Mesoproterozoic is that these fossil assemblages are low in eukaryote diversity, often just with a handful of species of eukaryotes. And then, beginning around 1 billion years ago, we see increases in the diversity in a lot of these eukaryotic fossil assemblages. And we also see, for the first time, fossils that we can fairly confidently place as in association with modern eukaryotic groups. So groups, clades within the eukaryotes. And Andy touched upon these earlier. We have Bangiomorpha pubescens, which looks strikingly similar to some red algae. Proterochlotis, which looks very similar to green algae. There are fossils, Orosphera heraldi, most recently published, that have a lot of characteristics that are found today in fungi. They may be related to that or part of a larger clade, fairly closely related. We also, in rocks a couple 100 million years younger, we see interesting biological innovations, the first evidence of biologically controlled mineralization. This is the work of Phoebe Cohen, where she described a whole diversity of apatite phosphate scales, which probably were something comparable to coccolithophores, coccoliths, although they're not. She's not suggesting they actually were coccolithophores back there. In younger rocks, some also phosphatized scales reported. And then it's kind of embarrassing because just to be consistent, I feel like I need to put in a picture here, but this work was done by another Knoll student. We see vase-shaped macrofossils in rocks 730 million years old that suggest, not only the presence of these structures, defensive structures, the test itself, but suggest the presence of yet another eukaryotic lineage, archalinid testate amoebae, which are part of the amoebozoa group of eukaryotes. Another important source of information for the early fossil record of eukaryotes, which Kliti touched upon, is biomarker compounds. So these are hydrocarbon molecules. They're derived from lipid membranes of ancient organisms. They get preserved in rocks, sometimes in modified form, and they are used to identify particular clades. So they're biologically, they're important, they're potentially taxonomically informative. And steranes here are the fossilized version of steroid molecules, and one example is shown here, these yellow molecules here, cholesterol, which get preserved as cholesterol, as Kliti noted. So sterols are so widespread in living eukaryotic lineages that it's thought that the last common ancestor of modern eukaryotes possessed these molecules as well. This diagram is modified from a review by Jochen Brox, and it shows a distribution of all these places where we've looked for biomarkers through time. And in these older rocks, you can see there are biomarkers present, but no detectable eukaryotic steranes. And then there's a gap, a couple 100 million years, and then it's not until just before 800 million years ago that we see evidence of eukaryotic steranes. So this record has been presented as something of a conundrum, because if you recall, the body fossil record that I was just showing you goes back more than 1,600 million years ago. And so it seems to conflict with that history, given that we know eukaryotes are much older. And in some cases, there are units like this Nonsuch Formation in North America where we have fossils preserved there. We have the hospitable environments, we think, for eukaryotes. They're oxygenated environments. And as Sarah Slotsnik and colleagues noted, it's a puzzle why these eukaryotic denizens preserved in the fossil record did not leave an appreciable sterane record. Now, the traditional answer to this conundrum is what I will refer to as the delayed diversification model. In this model, the last eukaryotic common ancestor, so this is the last ancestor of all eukaryotes alive today, which is referred to as LECA, was around at least 1.7 to 1.8 billion years ago. And so at this time, we had eukaryotic organisms that had all of those characters that I've talked about that characterize eukaryotes today. It had a nucleus, it respired oxygen, it was aerobic, it had sterols in its membrane, it had mitochondria, and it had a complex cytoskeleton. And yet, eukaryotes remained rare and restricted. Perhaps because they were aerobic, they breathed oxygen, they were limited to those patchy environments where there was enough oxygen around. And then, around 1 billion years ago, eukaryotes diversify. And this view is consistent with this idea that the evolution of complex life on Earth might have been modulated primarily by environmental factors through time. And in particular, one idea is that maybe eukaryotes were rare and restricted during this time because oxygen levels were really low. And then as soon as oxygen levels rose, or maybe its nutrient levels rose, eukaryotes diversified. And this explains this conundrum with the sterane fossil record, in that eukaryotes back here were making sterols, but they just were not present in any appreciable sort of abundance such that their sterols would get into the fossil record. Now, this model may be correct, and if I recall correctly, this is a model that I think originated with Andy Knoll, and as is usually the case, Andy's ideas turn out to be correct. So, I want to put this up there as something that I think may in fact be correct, but it's not the only way to interpret this fossil record. And what I want to suggest is an end-member possibility, sort of at the opposite end of this model, which can also explain these data. But before I talk through the model, I want to take a moment and talk about this concept of crown group, stem group, and total group, which has been touched on a little bit, but I think it's useful to go through in detail. So crown group eukaryotes are all of those eukaryotes that are alive today, their last common ancestor, which I've already mentioned, LECA, and all of its descendants, including extinct groups, like Tyrannosaurus rex is a complex eukaryote, crown group eukaryote. Total group eukaryotes include all of those lineages that branched off after the split with the last common ancestor, with the closest living relative to eukaryotes. As far as we know today, that's this group, the Asgardarchaea. And this is a group that lacks a nucleus, lacks a complex cytoskeleton, although it does have elements of genes related to cytoskeleton in eukaryotes. It lacks mitochondria. It's anaerobic. I can't remember if I already mentioned that. Right, so it lacks all of those characteristics that are in common in modern eukaryotes. And this guy here, this lineage, FECA, is the first eukaryotic common ancestor. So that's total group. And then the stem group is all those lineages that branched off before the crown group. And by definition, they're extinct. I think it's useful for me anyway to think about stem group and crown group and get insights by thinking about the record of dinosaurs, maybe in part because I teach a large dinosaur lecture, so I talk about these things all the time to my students. But here you see birds, crown group birds. That's the last common ancestor of all birds, Libca, last bird common ancestor, all of its descendants. Total group birds include all of those lineages that branched off after the closest living relative of the bird, so that's the crocodiles and kin. So pterosaurs, all these non-avian dinosaurs are in fact a type of bird. They are a type of total group bird. And in fact, all of those extinct forms form that stem group, that collection of stem group birds. And the cool thing about the dinosaur fossil record is that because we have such a fabulous, well-preserved record, we can actually see the sequence in which different characters that we associate with living birds, we can see the sequence in which they evolved. So we know, for example, now that powered flight evolved much later than hollow bones and feathers, which may have been present in the last common ancestor of dinosaurs. And so that means that feathers and hollow bones did not evolve for the purposes of flight, because flight came later. They were useful in that transition. It means that if we do see creatures that have powered flight, we can infer that they must have hollow bones, they must have feathers, unless they've secondarily lost it. But importantly, sort of back to this story I'm telling about eukaryotes, the presence of feathers in the fossil record, the presence of a creature with hollow bones, does not imply that that creature was able to fly. So applying this logic to the tree of eukaryotes, we can see that all of those characters I talked about that aren't present in the Asgard archaea are present in all crown group eukaryotes, unless it's secondarily lost. Nucleus, complex cytoskeleton, aerobic respiration, eukaryotic sterols, they all must have evolved back here, perhaps over a really long, drawn-out period of time. But because we don't have that great fossil record in dinosaurs, that the dinosaurs do provide us, we don't know the order in which these appeared. And so that means that if you see fossils like Geosciences and Tapania, which tell us, I think, convincingly, that a complex cytoskeleton was around, we can't say anything else about these creatures. We don't know that it had a nucleus. We don't know that they breathed oxygen. We don't know that they had mitochondria. And we don't know that they were making sterols. So going back with this in mind and going back to this simplified fossil record of early eukaryotes, I want to present this alternative end member to the delayed diversification model. Okay, sort of an extreme end member. So in this model, we know that FECA was around by the late Paleoproterozoic and maybe hundreds of millions of years earlier. I mean, the further back you get, right, toward this branching point with the closest living relative, the less likely we are going to be able to recognize any eukaryotic features. So some of those simple, smooth-walled, organic-walled microfossils I mentioned could, in fact, be part of the total group eukaryotes. We don't know. So we have FECA back here sometime. And these fossils here suggest that total group eukaryotes were around and that a complex cytoskeleton, or at least whatever was needed to make those spines, were around by this time. But let's say all of these are stem group eukaryotes. And then we go further in time, and we know by the time we see those recognizable eukaryotic groups, the red algae, the green algae, the testate amoebae, that the last eukaryotic common ancestor, the start of the crown group, what must have been around. So let's put it as late as we can put it, maybe 1150 or 1050, 1100, just before the appearance of this group. And this could explain this relatively late appearance of steranes, if in fact sterols evolved very late in stem group evolution. Remember, there's that gap for a few 100 million years where we don't have any sampling of biomarkers. And so maybe that's where the sterols appeared. And they're absent back here just because they hadn't been acquired by eukaryotes. Similarly, again, an end member possibility, maybe aerobic respiration is late as well. And that would imply that all of these fossils here are from organisms that were not doing aerobic respiration and did not live in oxic environments. So here again, I'll show these two end member models. And this time I've put in the stem groups in light blue and the crown group in dark blue. And the key difference really between these two models is when the crown group appeared, when LECA originated. In the delayed diversification model, LECA's back here, which means that at least some and perhaps many of the fossils that we have back here had all of those prime eukaryotic characters, most notably aerobic respiration. So they were dependent on oxygenated environments. In this model, however, LECA appears quite late. And so, for all we know, these eukaryotes didn't have many of the characteristics that we associate with modern eukaryotes. And the interesting thing about this model is it suggests that, rather than in this case where there was some environmental, perhaps some environmental constraint that kept eukaryotes limited through time, and then this sort of break was released and eukaryotes diversified. In this model, it could just be that eukaryotes diversified once sort of a particular innovation had been acquired, maybe aerobic respiration, and that's what led to their diversification. Of course, there's lots of options in between here, and I've just proposed this as sort of alternative working hypotheses, working models. I want to shift gears here quickly and talk a little bit, though, about what might have driven this diversification in the Neoproterozoic. And in particular, I want to focus on predators, the appearance of microscopic predators, and how it's possible they might have driven this diversification. I show you two examples here of types of predation that you see in modern protists. On the right is the ciliate Didinium. It's in the process of phagocytosing or engulfing another ciliate. And then on the left is a vampire amoeba, Vampirella, consuming the cytoplasm of a green alga by perforating the exterior, the cell wall of the green alga. Now, it's hard to overstate the effects that a predator can have on its prey. This is a chart that you've already seen in Andy Knoll's talk, where he talks about, he mentioned the drilling predation, or sorry, drilling intensity and through time. And you can see how it's correlated with the diversity of marine invertebrates through time, marine animals. And I should take a step back here and note, so the idea that predators are important for the diversification of their prey comes from some early work done in the 60s by an ecologist who did experimental plots and removed the top predator. This is in marine environment. He removed the sea stars. And what he found was that the diversity of the prey in those plots dropped. And the explanation for this is something referred to as the cropping hypothesis, where the presence of a predator prevents any single prey from getting too common. Then the absence of a predator, the prey compete with each other, and the most successful one will outcompete the others. A predator keeps the most successful one at bay, and you get some of the other prey sort of keeping a foothold. And this idea was built on by Steve Stanley in the 1970s, who suggested that maybe the diversification, the appearance of animals and diversification of animals was sort of the reverse of what pain did. Rather than remove a predator, if you add a predator to the system, the prey will diversify. As Andy already alluded to with the marine Mesozoic revolution, predators can also have a big effect on their prey by driving evolutionary innovation related to defenses. And this is an interesting thing we see in the early Cambrian, that a lot of the skeletons that appear to be adapted for defense. You can see, for example, in this chancelloride, which looks similar to a barrel cactus, although it's not at all related. It has these covers in spines. Halkyrid, covered in sort of a chainmail-like array of sclerites and 2 shells, seems to be consistent with this idea. And this is generally an argument that's been compellingly put forward by Stefan Bengtsson, who's in the audience, that the appearance of skeletons and the Cambrian diversification in general may have been driven by the appearance of carnivores, of organisms eating animals. This is a compilation from a project I've been working on for several years that is still in preparation, but the goal of that project was to look at the appearance of mineralized skeletons, independent acquisitions, across all eukaryotes, and see what type of mineral they evolved in what period of time. And this is just a sampling of that work. And the striking thing that you can see here is that out of the sort of almost 50 independent acquisitions of skeletons in animals, about 2/3 of them occurred in the early Cambrian. So I think this supports this idea that it's the appearance of predators, and you get lots of different lineages independently evolving defense. There's a hint of this going on in the Neoproterozoic when we first see eukaryotes diversifying. We see the mineralized scales appearing for the first time. That work of Phoebe Cohen et al., mineralized scales as well here. We also have organic tests, which are not mineralized, but possibly also involved in defense against predation. So the idea here is that although eukaryotes were around, at least 1,600 million years or more, at least total group eukaryotes, there wasn't anything that was eating them. And it wasn't until later, maybe around 1 billion years, that predators first appeared. And in the case this sounds weird, there is precedence for this. The first plants, for example, appeared not to have had anything eating them for 100 million years or so. Finally, somebody figured it out. Herbivores evolved. And the earliest animals also sort of lived in a Garden of Eden, where it doesn't appear that there were any carnivores around. And then in the Cambrian, carnivores appear, and you get that diversification. So what about eukaryotes? Well, this is a graph from a paper by Andy Knoll and Dan Lahr, who tried to reconstruct the feeding mode across eukaryotes. And the black color here is eukaryovory, so eukaryotes that eat other eukaryotes. And you can see that ancestrally it looks like eukaryotes were bacterivorous. They ate bacteria, most likely, or were osmotrophs, so feeding on dissolved organic carbon. Let's see, I'm running short on time, so I just want to quickly go through and say, phagocytosis is hard to preserve in the fossil record. I don't know that we'd have any hope of finding that. But just quickly, I wanted to point out some evidence that I found a few years ago for evidence for drilling predation in eukaryotes. And that comes from fossils from the Grand Canyon, rocks that are 770 to 730 million years old. Here are these organic walled microfossils. I was doing a systematic study of them, and I started looking at them under the microscope, the scanning electron microscope, to look at their morphology. And entirely serendipitously, I started noticing that some fossils have these little holes, these perfectly circular holes, some of them appearing kind of beveled. So here's a number of them scattered around. And you can see a little bit of this beveled nature of them. If we do a TEM image and cut a cross-section through, it looks like that it's perforating from the outside. Here's another example. They were found in a lot of different species. They weren't common in any single one species. And they looked identical to, fast forward, they look identical to marks made today by modern vampire amoebae in fungal spores and in green algae. And this is what they look like. They can be a real problem in algal ponds where they're growing biofuel. They can decimate a population. And the vampire is an apt name. You can see here's a fungal spore, and the amoeba here. It's eroding a circle, circular mark. It lifts up the plate like a manhole cover and swallows it, and then it inserts its pseudopods inside, and it takes chunks of the prey's cytoplasm. So I think that's what's going on with those organic walled microfossils. I went back and looked at those vase-shaped microfossils that I had studied while a grad student at Andes, and noticed that there are circular holes, quite larger, and also these semicircular holes that tend to be arranged with the long axis parallel to this flat part of the semicircular hole. Suggesting maybe these were made by predators that had a stereotype behavior and arranged the test in a certain way. In the Chuar group, there's actually a lot of evidence for different types of predators. There's these tiny very tiny holes, 0.1 micron in diameter, the larger circular holes, the semicircular holes. The testate amoebae themselves are known today to be predatory. And then also some biomarker work from Jochen Brox, which suggests a presence of a toxin that would have made the organism poisonous to being eaten. So just to conclude, I like this idea that, that maybe the arrival of eukaryotes, of predators on eukaryotes, might have driven the diversification, in that it's sort of an earlier case that we see later in the Cambrian diversification of organisms, that the appearance of carnivores here is a reprise of the appearance of eukaryotes here. And I'm fascinated by the fact that we tend to see these repeated patterns at different scales in the history of life. We see the evolution of camera-type eyes in protists, and then a comparable structure that is being reprised by multicellular organisms here in the squid. There's another great example here where you have two related organisms, the jellyfish and the Portuguese man-o-war, but in one case, this is a single animal. The Portuguese man-o-war is a similar form, but it's a similar form that has evolved as a result of evolution of a colony of animals. So sort of like a super organism. Okay, so sorry I've gone over, but here are my conclusions. Basically, the takeaway is that we have these fossil data, right, which provide us with an outline of the early evolution of eukaryotes. And this model, the delayed diversification model, has been sort of the dominant paradigm that's out there. And it may be right, but there is an alternative way to interpret the data. And I think the important point here is the takeaway is that eukaryotes were not necessarily aerobic way back here, and it may not be that oxygen was controlling their diversification. Predators may have been the big innovation that drove the diversification of eukaryotes. And the things we see in animals, the patterns there, are often just a repeat of the protists who did it first. So that's it. Thank you. And thank you, Andy.